Hello everyone. Thank you for listening to our presentation. First, I'd like to take this opportunity to thank Gard for organizing such a wonderful event to share the work that we did with detection of bacterial pathogens in low moisture foods with direct and ramen mapping with you. So this work is done in my lab. My name is Dr. Chenzu Yu from Iowa State University, and Cheng Pan and Binmin Zhu are two grad students working with me on this project. So first, I'd like to talk a little bit about the motivation why we did this work. Because low moisture food are the foods with a water activity less than 0.85. They used to be considered pretty safe, right? Like peanut butter, black pepper powder, milk powder, and the egg powder. They were considered safe as their water activity is so low so that microbial growth is inhibited in it. However, lately that we have realized that these foods are not actually as safe as we have hoped. And certain pathogen species like Salmonella, they can survive in the low water activity environment for a relatively long time. So they still pose as a health threat to consumers. In foodborne diseases outbreaks related to low moisture food like peanut butter, we all saw that is increasingly being reported. So it does pose as a public health threat. And conventional detection techniques have limitations that render them less effective for low moisture food. I will talk more about this later. So, and here is a snapshot, and over a 3-year span, there are over 1,000 foodborne disease cases were attributed to peanut butter or peanut butter containing product. So we can see that the contamination in low moisture food is a problem that we have to address. By far, the most important species that pose as a threat in low moisture food are salmonellas. From this table that we can see, salmonella is the most significant threat. And so our work here, salmonella is one of the model bacteria that we chose to look at for our detection research. So conventional detection methods like culture-based methods, molecular methods like ELISA or PCR, these are all widely used for pathogen detection in food. But when it comes to low moisture food, there's some limitations. Culture-based method is time consuming. It takes a few days for you to get your result back. ELISA and the PCR both are designed for analyzing aqueous samples, right? So for people to use them on low moisture food, typically we'll need to wash the food and analyze the wash water. Low moisture food cannot be directly interrogated with these molecular acids, which limits their application. Because wash water, although we can work with wash water, but wash water is an indirect material for us to detect pathogen for low moisture food, which also means that you cannot use these methods to conduct any inline detection for food directly. You have to get samples and wash the samples and analyze the wash water, right? So that's why that we wanted to develop something that can work directly on low moisture food. The scheme, let me move this out of the way. So the scheme that we developed, we give it a name called Dual Immunological Raman-Enabled Cross-Checking Test, or DIRECT. So here is how it works. We have a probe, molecular probe made. And this molecular probe can bind specifically towards a pathogenic bacterial target, for example, E. coli or Salmonella cell. Right? E. coli and Salmonella cell themselves can be characterized by their Raman signature, but Raman scattering is a weak phenomenon. So when you have a few cells embedded in a large quantity of low moisture food, this cell signature can get overwhelmed or hidden behind the strong low moisture food background signal. Okay, but with our Raman probe, when we apply the Raman probe, they bind to the cells specifically through the molecular recognition reagent that we put on the probe surface. And then binding of the nanoprobe will induce a phenomenon called surface enhancement Raman scattering, which will enhance the signal coming from the bacterial cell and the probe all together. Then in the spectrum that we measure from these contaminated samples with the probe bound to the cellular targets, now we can recognize in theory the peaks that are coming directly from the probe and from the microbial cell. And by looking at or by identifying these dual signals, that we can give a positive identification or detection result for the pathogen cells. So this is a scheme, how this direct method works. The foundation of this direct method is surface enhancement Raman scattering. So when we have nanostructures get into close distance to a cellular target, then the interaction between light and the molecules in the cells can be enhanced by the plasmonic resonance coming from the nanostructures. And this enhancement can increase the signal intensity of the bacterial cell, which would be like undetectable to a much higher level so that they can become detectable. And this surface enhancement effect is very much dependent on the distance between the nanostructures and the cells. So only when the cells and the nanostructure are in close distance, less than a few nanometers, mostly less than 10 nanometers, then this enhancement can be significant enough for us to detect a sizable change in the signal intensity. In theory, the enhancement can be as high as 10 to the power 13 to 14, but in reality, we're more looking at 10 to the power 4 to 10 to the power 6 at the most. Right? But the beauty of this is because the probes and the cells needs to be in close contact with each other, so only when molecular binding events occur that we will detect that dual effects, the dual signals coming from both the probe and the bacterial cells. So which means only when specific binding happens, then we can see this dual signal. If your probes are not binding to your cells in the system, then you don't see this dual signal. So which means that binding events can be detected under this scheme. And given if we use the molecular recognition reagents like antibodies that can specifically bind to a target, then binding events means it gives the specificity that we need for detecting a specific bacterial target, which is the foundation of this approach. Then in our work, we use both portable Raman spectrometer that can interrogate a sample very fast, very easily. And this will allow us to bring the spectrometer into field to do on-site or in-field detection, and a Raman microscope, which allow us to do high throughput Raman mapping for increasing the detection capacity or throughput of detection. I'm going to talk both of these later in my presentation today. So the probes that we used are made in our lab. We use a gold nanorod for the nanostructure to give us the enhancement effect. We use a specific design of developed chemical conjugation procedure based on 4-ATP, also developed in our lab, to link the MRAs, the molecular recognition reagent, to the nanostructure to make a Raman probe. And MRAs that we investigated are antibodies, typically for molecular recognition work, and peptides provided by our Chinese collaborators. There are two peptides that we used, which both can specifically bind to Salmonella cells. And we looked at two model food systems, black pepper powder and egg powder. And we also looked at two bacterial targets, E. coli and Salmonella, as our model bacteria for our detection tests. So here shows the kind of things that we, the nanoprobes that we made, right? So going from a nanorod to ATP conjugated nanorod to antibody and a peptide conjugated nanorod. And through this process, we take a Raman spectrum measurement by looking at disappearance or the appearance of specific bands or peaks associated with each of these chemical steps that we can establish or we can validate conjugation chemistry has been successfully implemented in this process. So here shows some of the results that we obtained with E. coli as our bacteria model and with black pepper powder as our model food. I know that by just looking at the spectrum, it's not that easy for us to see, okay, these are different. Because the black pepper powder gives a very strong background signal. So you can see that the signals are pretty much dominated by black pepper powder. But by close investigation of the samples spec with and without E. coli, which is, let's see, this is with E. coli. And let me see, PP, this is without E. coli. Okay, so these two are with E. coli, without E. coli. Then by looking at the spectrum features, we can actually see peaks that we can identify with a certain chemical structure in the probe, in the antibody, and in the E. coli cells that show some difference. And these results are obtained for a spike level at 10 to the power 3 CFU per gram. This is not per milliliter, this should be per gram here, right? And certainly this is encouraging, but to really look at the difference in the spectrum features that we can identify with and without E. coli contamination, we need to conduct discriminant modeling. Some statistical analysis on the spectrum data that we have collected. And we also tried this with egg powder, a different type of food. And the egg powder is more difficult than black pepper powder because egg powder are rich in lipids and proteins, which will tend to be more overshadowing than black pepper powder because it have more overlap with the cell signals. But still, by conducting discriminant analysis down the road with a statistical analysis, so we can get pretty decent discriminant or differentiation results between with and with E. coli egg powder samples. So we did PCA first, and then we conducted a support vector machine differentiation analysis. So within the first 5 PCs, the 1, 3, and 4, I mean, we can hear this is the first PC, second PC, and the fourth PC. Right? These 3 PCs contain information that give us the power to distinguish between samples with E. coli contamination and without E. coli contamination. And for different concentrations as well, for different spec levels. And these results are for spec levels at 10 to the power 2 CFU per gram, and CF 10 to the power 3 CFU per gram. So what we can see is differentiation or the separation between with and without samples are well established for the spectrum data that we have collected. And we also looked at further than when the concentration or the contamination level changes, whether that will change the accuracy of the differentiation analysis. It turned out for black pepper samples, when the contamination level is at 10 to the power 2 CFU per gram, we can still reach about 94% accuracy in the differentiation analysis, which is pretty good in terms of telling samples with contamination versus the one without contamination at 10 to the power 2 CFU per gram. It's not too bad. And for the contamination level get to 10 to the power 3, this accuracy rises up to 97%, which is actually very decent. And for egg powder, well, it's not as good. As I mentioned earlier, egg powder is a more difficult sample to work with. So we're still in the process for further improve the detection accuracy there. And the bottom line is, with this technique that we can detect with confidence 10 to the power 2 CFU per gram level of contamination in black powder, the black pepper powder contaminated with E. coli. And then we looked up at a different MRA, which is peptides. Peptides are shorter protein, chains, right? So these are much smaller than antibodies. And as I mentioned, the surface enhancement is dependent on the distance. So when the linkage molecule, the MRAs become smaller, then more enhancement is going to be introduced to the cells. So peptides, in theory, should be better than antibodies. And we looked at 2 different peptides. These are specifically selected based on literature for targeting salmonella. And these peptides are provided to us by our Chinese collaborators. And the results are, again, pretty promising, because look at here, this is with salmonella, this is without salmonella. Clearly that we can see some features are changing with and without salmonella, right, when the probes are being applied to the samples. This is black pepper powder. And with the second peptide that we have tested, the result becomes even better, because now we can identify specific peaks that are associated with this detection scheme. The results are, again, really good, because these contamination levels here are both at 10 to the power 2 CFU per gram. And based on this, we developed a high throughput Raman mapping technique. So what we're doing there is that we have a tape that have 25 pixels, right? And in each pixels, you have different sample. For example, if you have 4 samples, 1, 2, 3, 4, which are contaminated with a bacterial target, in this case, salmonella, and upon applying the probe to the surface of this tape, and then we do a Raman map of the surface, for the pixels that you have contamination, you expect to see both the probe signal, as we show here, 1, 2, 3, 4, and the bacterial signal, 1, 2, 3, 4, and the bacterial signal at a different peak position, 1, 2, 3, 4. Okay. And in theory, you can look at the peaks that are associated with this probe bacterial binding and generate multiple of this map. And if these 4 spots are showing up positive in all of these maps, then you know for sure that on those spots that you have contamination. And with this, we can acquire a map to analyze 25 samples from one little tape. So we did this with some test runs. So here you can see 3 peaks, 2 are salmonella peaks. So 1 is a probe peak on 3 maps that we created by scanning a tape surface. And clearly we have this spot showing up on all 3 maps as positive, means that at that spot we have a contaminated sample. Right? So this shows the potential of using this directly on low moisture food samples for quick screening of pathogen contamination. And as a conclusion, and my time probably is running operated as a conclusion. Direct can perform rapid pathogen detection on different low moisture food with relatively low limitation of detection. Using target-specific peptides and mRNAs can further improve the accuracy and the reproducibility of the Direct scheme. And with Raman mapping, location of the pathogens can be detected in powder. I apologize for the spelling mistake there. In powder foods, which could be a significant improving sampling and throughput of this detection scheme. So we're still working on further improve this scheme, and hopefully that we have more to report down the road. And I'd like to thank USDA ARS for partially funding this research, and my collaborators, Dr. Byron Brunstad from ISU for providing us with the microbial samples that we used, and also Dr. Sun Na and Dr. Liu Xiaoyang from our Chinese collaborator, Dalian Polytechnic University, for providing us with the peptides that we used as mRNAs. So now it's time for questions. Thank you for listening to our presentation.